If you look at us on the outside, we're pretty symmetrical, but if you look on the inside, we're incredibly asymmetric, and the most asymmetric thing is our heart. Early in development, you have a head and a tail and a front and a back, but you don't have a left and a right, and you need the embryo to have this internal reference. Cilia are these little structures that are stuck on the top of every cell. So what the embryo does is it takes these very asymmetric cilia and positions them on a group of about 250 cells that come up at about 18 days of human gestation. And now those cells have their own internal reference. Oh, I am asymmetric. This is my left side. This is my right side, because the cilium on top of me points the way. And that mechanical signal is then transmitted to the whole rest of the embryo, including your heart, your lungs, your liver, your gut, et cetera, to tell them, oh, this is left and this is right. So we're part of a national consortium that is essentially trying to build the largest congenital heart disease database. We have recruited a total of about 17,000 patients. And what we're trying to do now is link that genetic cause to a developmental mechanism that makes the heart develop improperly. So my lab is focusing on cilia, which we've turned up in the genetic analysis also as a significant cause of human congenital heart disease. We're trying to see how they impact not just whether your heart's on the left or the right, but also they might impact heart function. We know that they impact early valve development, and those are places where you could imagine that you could have some potential to intervene and make things better. We go from two angles. The first angle is the large-scale genomic studies. So a lot of our work was supported by Yale Center for Genome Analysis. And so one aspect works through the geneticists and the more statistical genetics approach to defining what the contributing causes are. And the other aspect, we work with models to try to better understand how the genes we identify in humans actually function in heart development. And we're trying to determine at what point in development the heart has its very earliest structural left-right asymmetry, and how those early signals from the ciliated cells in the left-right organizer communicate to the developing heart tube to make it go left or right. Our hope is that we can first really understand the genetic architecture of congenital heart disease on one hand, and then understand how the heart develops, especially in its most early steps, to understand the molecular mechanism that drives heart development. And understanding that will give us some cues as to which patients might respond to what therapy. I mean, we can't make the heart loop in the other direction because that happens at 20 days of human gestation, but we can help deal with the outcome postnatally.